Thank you for having me. So today I'm going to share with you our study on combating neurodegeneration. So I'm part of a research group here in biology, study the health and disease of the nervous system. The nervous system is a command center, basically make up who we are. And the health and functionality of the nerve system depends on the health of neurons, neuronal network, and the whole environment housing the system. And we study neurodegeneration, which is basically neuronal loss, and depends on where the neuronal loss occurs and which type of neuron that's being affected. Neurodegeneration can lead to a wide range of symptoms and diseases. For example, if it's the motor neuron that's being lost from the motor cortex or spinal cord, the outward display will be the motor neuron disease, amyotrophic lateral sclerosis. If it's the dopaminergic neuron that's being lost, the outward display will be Parkinson's disease. The same thing, if it's the frontotemporal lobe that's undergo degeneration, you will have frontotemporal dementia. So basically, neurodegeneration comes in all forms and leads to all kinds of different types of disease. Depends on the where and what neurons being affected. However, despite this huge variety, neurodegenerative disease do share one common feature. That is the accumulation of abnormal protein aggregates, as seen in the dark staining here. So now the questions become, are those abnormal protein aggregates responsible for neurodegeneration? And if they are, can we target them for disease therapy? So that is the question today. I'm going to focus on the protein TDP-43 in motor neuron disease as an example. Here, is TDP responsible for neurodegeneration? This is a study conducted by an American group, and they use the transgenic technology to turn on or off the expression of TDP protein shown in green. So you can see the first figure is a control situation where there's no abnormal protein expression. Everything's fine, the neuron is intact, the animal's healthy, no problem. What happens is they then turn on TDP expression for 6 weeks. You start to see these green proteins accumulated inside the cells. As a result of that, we have a reduced number in motor neuron. So the dotted line is the count of motor neuron you are supposed to see from a healthy animal per spinal cord section. And that count reduced after 6 weeks of protein expression and accumulation. And what it means to the phenotype? So for motor neuron disease, patients suffer from motor function decline and also muscle weakness. And that is what we see here. So look at the muscle strength and the motor function. Both decline steadily within the 6 weeks of protein aggregation. So this is showing that the presence of protein aggregates indeed causes the loss of neurons and then the functional decline. What happens then is they then switch off TDP expression from there, and unsurprisingly, the neuronal count is not recovered because neurons are not like our blood or skin cells. They do not regenerate. So what you lost. However, the functionality can be recovered. So this means that if we can find a way to stop piling on this horrible protein onto the neuron and just give the surviving neuron some time to recover, despite we cannot recover the lost neurons, we can recover the functionality. And therefore, if we can find a way to target those horrible aggregates, we may have a therapy. Then how do we do that? This is a very hot research area a lot of groups are working on. In our group in York, we focus on one system that the cell is supposed to be using already to deal with protein folding and refolding. That is the chaperone system. So the chaperone system is made of by a bunch of heat-sharp proteins, and they should work together to facilitate protein folding, refolding, and degradation in the cells. The idea is that the chaperone system has some kind of basal activity in any healthy cells, but when the cells are faced by stress, like temperature change or the accumulation of abnormal protein, this whole system should be activated and tackle those aggregated proteins to help the cell get over it. And this is a simple illustration. The chaperone response are governed by a master regulator called HSF1, normally bound by HSP proteins. During stress, those HSP proteins will be removed from HSF1, allowing the regulators to trimerize and move into the nucleus. Yeah, where they should then activate the downstream expression of HSH genes, HSH protein genes. And the upregulation of the HSH protein genes will then tackle those protein aggregates. Okay, so there, in the best case scenario, they should bring those protein aggregates into refolding, i.e. to transform the structure of those protein aggregates from those horrible aggregated structure to the properly folded functional structure. Failing to do that, those chaperone protein can also bring those aggregates into the degradation system, basically to get rid of those aggregated protein. So that should be what's happening in the cells every day to deal with proteins, any proteins that's unwanted or misfolded. So our task is to study whether we can engage this system and make the system to deal with these horrible protein aggregates that's causing neurodegeneration. So the way to do that, we use the cell line and express different versions of HSF1, the master controller. We have either a version of HSF1 that will activate the chaperone activity very well, and one version that will inhibit the chaperone activity. And as you can see here, if we activate the chaperone activity, the level of insoluble protein TDP aggregates are remarkably reduced compared to control. And that is an activation of chaperone activity compared to that. So it shows that it works very well. Once you activate the chaperone activity, you reduce the amount of protein aggregates. And not only that, the activation of the chaperone activity also reduces the toxicity to the cells. So the expression of TDP per se will cause cell death. As we see, it causes neurodegeneration, basically. If we activate chaperone activity, the level of degeneration will be reduced. So that's a very useful system and should be working. Then what happens in the neurons in patients? Why the system is not functional in neurons? So we take a look of what happened in disease. This is a mice study. We look at the protein level of HSF1 and chaperone proteins and see what happened in the mice model of ALS, that is this one. You can see that the protein level of those crucial chaperone proteins are already significantly reduced in the animal model of ALS, which is validated in human patient data. Again, the chaperone proteins are significantly reduced in patient of ALS compared to healthy control. So overall, this data shows that the activity of chaperones somehow is compromised in ALS and probably contribute to the disease progression. However, if we can find a way to re-engage this chaperone activation, there's a chance we can refold TDP and rescue the neuronal death. So the question becomes, how do we do that? In our group, we tried two approaches. The first one is the gene therapy approach, which basically we inject the gene of HSF1 directly into the brain, in this case, the mice brain. So this is the expression profile we have when we induce this gene. Basically shows a nice expression across the brain. And the result is here. I want to focus on what happened in the TDP mice. Again, we are looking at motor function and high-link strength. You can see that in both cases, the animal express HSF1 shown in the orange line shows a better performance in motor function and also handling strength compared to the animal of control. And this is not a magical rescue, but quite encouraging, given that we only give the animal 1 dose of injection 4 months after the disease onset. So promising. However, gene therapy is not everybody's cup of tea. So we do look into another avenue as well, which is the new drugs discovery. So what we want to do is we want to re-engage this whole pathway via HSF1. How do we do that? We know HSF1 is negatively regulated by another protein, HSP90. So what we eventually want is to find a new chemical that will disrupt this interaction and therefore release HSF1 to activate the chaperone response. So this is the system we use, basically by measuring the fluorescence protein, one luciferase protein and one fluorescence protein to gauge the interaction status of HS1 and HSP90. I'm not going into detail there, but basically this is a study collaborated with the drug discovery unit in UCL. And this is our first ever chemical compound screening. And you can see that the blue dots represent the negative control. So anything along this line indicates there's no impact on this interaction. And red dots are our positive control. So whenever there's a compound impacts on this interaction, it will reduce the ratio of input-output signal. So in this very first chemical drug screening, we actually identify, first of all, one known chemical that will interfere this interaction. That is great, because we identify that blindly, indicating the whole system is working as it should be. And more excitingly, we do identify novel compounds. So potentially, this is a new compound that will interfere the interaction of HSF1 and HSP90, thereby activating the chaperone response. So this is a work in progress, obviously. We are currently expanding the screening to include a larger library and also following up the validation of any hit compounds. Fingers crossed. So overall, in today's talk, I hope to convince you that neurodegeneration is very wide instead of its symptom and the disease displayed. But fundamentally, they are characterized by the cells failing to deal with protein aggregation. So how can we help the cells alone to deal with protein aggregation? In our study, we focus on the chaperone system and trying to find a way to re-engage the chaperone system, either through gene therapy or through a new drug that can activate HSF1. And by doing that, hopefully to help the neuron to deal with protein aggregates and recover its functionality. And all these works are carried out by my staff, students, colleagues, and myself at KCL and York, with a wide collaboration with everyone supporting it, MRC, Wellcome Trust, MNDA, and Peterson Mill Trust supporting our research. Thank you very much.